Hi class, welcome to our 13th lab, lucky number 13 lab for this semester. And this lab is going to focus on two labs that exemplify the relationship that some of these pathogenic microbes have with the host, specifically us in a human host. So this lab focuses on lab 521, which is the blood hemolysis lab, and 523, which is the coagulase lab. 521, there's no questions that you have to do. 523, I like question number 3 in the data analysis, but again, really no data to interpret on this. And the quiz is super hard, super hard. So I recommend taking the quiz right after you hear this lecture so that you can remember all the tiny little details. I hope you catch the sarcasm in my voice right now. But I would get this quiz done right away just so that you can take care of it and get it done and out of your hair as soon as you listen to this lecture. So let's go ahead and dive into the introduction to this lab and the idea behind this lab. And if you remember in Unit 4, in the, what would that be? I think it's lecture maybe 16, we talked about how some bacteria basically can fight back against host factors that limit their ability to survive or grow inside the body or on the body. And one of these virulence factors or pathogenicity factors is the secretion of toxins. So remember, if it's secreted, is it an exotoxin or an endotoxin? Ask yourself if you can recall. This is one of those kind of fundamental things that you should take with you from this class, is the difference between an endotoxin and an exotoxin. If you recall, an endotoxin, there's only one, and that endotoxin is LPS, and that's that lipid, kind of outside lipid barrier on that outer membrane of gram-negative cells. But most toxins that bacteria have are actually exotoxins that they secrete. And these toxins can have varying levels of toxicity. So for some of these toxins, a very small amount. If you recall, we talked about the tetanus toxin and the botulinum toxin. They're very toxic in very small amounts. But for the two that we're talking about today, they're not quite as toxic as those, but they certainly can cause harm to the host, and they allow survival of these bacteria inside the host as well. So the advantage to these, if you want to call it an advantage, the advantage to these these exotoxins is that we can characterize bacteria based on what toxins they have. So, for example, we know that certain bacteria express certain exotoxins and other bacteria don't. So if we know this, we can kind of help identify bacteria based on what toxins they're secreting. So in lab 521, we're going to look at how bacteria behave on blood agar. And we actually haven't used blood if we would have been in lab in person, we would have used blood agar before. But this it's kind of a cool agar, one, because of the presence of sheep's blood. Well, let me back up for a minute and talk about what blood agar is. So it's basically tryptase soy agar with a supplement of about 5% by weight sheep's blood. And essentially what that does is it turns it from a buffet, like your garden variety everyday buffet, to a really good buffet. And if you recall when we were talking about like fastidious versus non-fastidious, a lot of bacteria, and in fact a lot of bacteria that are pathogenic and that really like growing in us and humans, are actually pretty fastidious. They need a lot of these supplemental factors in order to survive, and that's why they like being in us so much, is because we provide those factors to the bacteria. So adding this sheep's blood to the agar is going to allow us to grow bacteria that are actually pretty picky, and I shouldn't say picky, pretty needy, and need a lot of these kind of unknown growth factors that blood, in this case sheep's blood or our blood, can provide. So just generally speaking, blood agar is really good for growing fastidious bacteria, but it also does something else for us, because we can use this type of agar to differentiate between bacteria that secrete these exotoxins called hemolysins. And hemolysins are enzymes that bacteria can secrete that basically destroy red blood cells. So they destroy erythrocytes in our blood. And of course, you can imagine that if this is an active infection in a person, this can have really deleterious effects, because it can give us anemia, and it can give us, I can't recall what the name of it, but they basically a low red blood cell count, which can lead to anemia and other issues in terms of like basically a hypoxic environment, right? If we don't have red blood cells, we are not getting oxygen where it needs to be in our body, and that can cause problems. So how does this actually help the bacteria? Like, why would a bacteria knowingly hurt its host? Well, I don't think it knowingly does anything, right? But what happens here is that if you recall, one of the innate immune responses to a bacterial infection is to try to gobble up or sponge, all of the available iron up, whether that's in our blood or on our skin or wherever it might be. Our body kind of has these global switch factors that want to sequester iron away from bacteria because bacteria need iron. It's a very essential cofactor to a lot of enzymes, and if bacteria can't get iron, they don't do well. They can't perform a lot of metabolic activities that they otherwise could. So the idea here, and this is actually still kind of unknown. There's not a lot of really good um, this is how would I say this is hypothetical. This is what scientists think the reason for this. We really don't know. We don't know what the big advantage is to this. But the idea here is that these hemolytic enzymes cleave open and break open these red blood cells. And of course, we know that red blood cells are full of iron, right? They have lots and lots of iron in them. And so the idea is that if they break open these red blood cells, they're basically kind of breaking open a loot box and getting all that iron that we've been withholding from them. So it's kind of a cool idea. Again, just that classic host microbe kind of battle. We're trying to keep iron from them to kill them. They're killing our cells to get it for themselves and so on. So there's 3 different types of hemolysin that bacteria can have, and especially Streptococci. They're really the main family that we're looking at when we want to do this hemolysin test or this blood agar hemolysis test. So the 3 possibilities, first we have beta hemolysis. And what this means is in beta hemolysis, or when a bacteria secretes beta hemolysin, it completely destroys the red blood cells. And you can see this so there's bacterial growth here throughout this streak. And what happens is the media essentially turns clear. It almost looks like it's your typical tryptocase soy agar, and it's just kind of that pale yellow agar looking color because the red blood cells are completely destroyed. Now, on the other hand, we have alpha hemolysis, which is, of course, carried out by alpha hemolysin. And this is partial destruction or incomplete destruction of the red blood cells. And what we see here is this kind of weird, kind of greenish olive halo around the bacterial growth. And you can't see, just so we're clear, there is really, this isn't the best picture, but there's substantial growth here. And you see there's a halo around this growth. So there's definitely bacteria on this. You can see the bacteria clearer in the alpha hemolysis just because the background's a bit darker. So there's nice bacterial growth on all 3 of these plates, but here you just see that really large yellow halo, clear halo in beta hemolysis. But whereas in the alpha hemolysis, you see this incomplete destruction of red blood cells, and that gives a really kind of green olive halo around the bacterial streaks. Finally, we have our last one, which is super confusing, and it's kind of irritating to me because gamma hemolysis actually means no hemolysis. No destruction is taking place. So when we talk about gamma hemolysis, it's really no hemolysis. And it's kind of annoying that they term it this because it's not happening. But you can see this because there's no halo, right? The red blood cells are intact. And really what you just see is bacterial growth on the surface of the agar, just like you were growing it on any other agar. So based on this kind of clearance pattern around the growth, so either complete, incomplete, or none, we can characterize the type of hemolysin that it has, and this helps us differentiate between the different strep species. So, excuse me. In lab, we don't. I can't show you any results today because what we typically do, we don't want to work with these bacteria. These are, again, bacteria that have these hemolysins, at least alpha and beta, are pathogens, are known human pathogens, and we would rather not work with these in lab. Simply because the threat of getting a contamination, having an active infection is too high, and there's no reason to put students at risk for this. So what we usually do in lab is we do a throat swab. We learn how to do a throat culture and a throat swab, and then we streak for isolation onto a blood agar plate, and then in 48 hours we see if anyone had any alpha or beta hemolyzers in their throat. So we don't have any results to record. And then again, I just want to briefly talk about the application here. This is a really nice application. If we were doing our unknown, for example, we would be able to differentiate between our different Streptococcus species based on their hemolysis pattern. All right, so let's go on to our second lab. This is lab 523 and the coagulase test. Again, as always, we're always testing for the presence of a particular kind of enzyme. In this case, we're looking for an enzyme called coagulase. And coagulase has two different forms. It can be free. In other words, it's a true, I shouldn't say true. It's a protein that is secreted into the external environment of the bacteria. Whereas the bound form is actually stuck to the outside. It's the same enzyme. Just one is free floating in the kind of media or environment surrounding the bacteria, whereas the other is stuck to the surface of the bacteria. And the reason that coagulase is considered, so coagulase actually does something really cool. What coagulase does is it takes clotting factors from our blood, right? If you take out the plasma from our blood, we have different types of clotting factors that essentially aid us when we get a cut, for example, right? And that's what causes a scab and so on. So what coagulase does is it hijacks these clotting factors to literally create a fibrin barrier around the bacteria itself. So it's basically cloaking itself in human proteins, right? And this does a couple things. The big thing that it does is it's basically coating itself in human antigen. So that's a big, how would I say? It's basically like a cloaking device, right? It's hiding from our immune system by surrounding itself in human antigen, or at least self-antigen, so that our immune system can't see it. This also helps, just the fact that it's coating itself in these fibrin strands also prevents phagocytosis. It makes it harder for our phagocytic cells to engulf these bacteria. So really big pathogenicity factor. Staph aureus, Staphylococcus aureus is among many, many tools. That Staph aureus is a big problem for people. Most of you probably know it because of its antibiotic resistance. If you've ever heard of MRSA or VRSA, MRSA is methicillin-resistant Staphylococcus aureus, and of course VRSA is vancomycin-resistant Staphylococcus aureus. We actually had a suspected MRSA case at I think it was at Mesa, and I want to say that student came to our gym once. We had to close our gym down for like 10 days and pay an environmental company like a ton of money to come in and decontaminate the entire gym, every surface, every nook and cranny of that gym, because there was just the slimmest possibility that that MRSA Staphylococcus aureus bacteria was in our gym. Big deal. And Staph aureus has just a lot of pathogenicity and a lot of virulence factors, and this is one of them. So it's definitely an interesting bacteria to study. So the way that we can tell if a bacteria is coagulase positive or not is to inoculate it. Believe it or not, so the recipe isn't really even a recipe. It's just simply rabbit plasma. So we take rabbit blood, separate the plasma from the cellular factors, and we inoculate it with our bacteria. And there's two different tests that we can do to determine whether or not coagulase is positive. We can do a tube test where we simply grow, so that's the bottom one here, we grow the bacteria in a small tube of rabbit plasma, and the tube test can test for both forms of coagulase. It tests for free coagulase or there's no space here. Can test for free coagulase or bound coagulase. So this tests for both. Whereas the slide test, essentially what we do is we we put a drop of the rabbit plasma onto a glass slide, and we look for clotting to occur. And this happens, the slide test only tests for bound coagulase. So, and this, I think we wait like 10 minutes. We don't typically do the slide test in class. We usually only do the coagulase test. But it's important to note, especially for the quiz, the difference between these two tests. So we already talked about how this helps bacteria. So we're good on this one. And then we have the results. Again, if we were in class, we would test a known positive and a known negative. Staph aureus is coagulase positive, and you can see that it clumps it basically kind of turns it into like a gelatin type. It's like a big clump, because it's activating again those clotting factors that are contained within the plasma. And Staph epidermidis is coagulase negative, and so it looks just like you would expect media to look like. It's like essentially like water, a liquid portion. So there are your results for the coagulase test, and that does it for this lab. Nice short lab. I thought about not including it, but I wanted to keep the points and give you an easy quiz. So again, be sure to pop over to that quiz and take it right away, and you'll be able to finish that and get it done, and get it out of your hair. Good luck.